In the M&M lab and with the cricket cartoon, we saw how differences in fitness could cause evolution to happen or a population to change over time. But there's other ways that evolution can happen as well. So by the end of this video, you should be able to describe 3 other methods of evolution. We're going to also get into some of the complexities of natural selection, so you should be able to compare and contrast natural selection on single genes and on polygenic traits. So just to review some important things to remember, that evolution is a change in allele frequency over time. So our gene pool, or the collection of alleles in a particular population, has to change over time in order for evolution to be happening. And natural selection itself is just one method that evolution can occur by. And natural selection requires 3 things: genetic variation, limited resources, and variable reproduction. So in this example with the bear, who has apparently learned a lot of important things, we've got genetic variation. So first of all, there are some bears that are smart and some bears that are not quite as clever. They can't get the garbage can lid off of the garbage. There's limited resources. The food is in the garbage can, and not everybody can get to that food. There's not enough food to go around, and so there's some phenotype that provides a advantage. In this case, it's intelligence, and so those bears that have the intelligence, that are fit, they're going to survive. They're going to reproduce, and they're going to pass their genes for their smartness on. So natural selection can happen on 2 different types of traits. Some traits are caused by single genes and other traits are caused by many genes. So when a trait is caused by a single gene, one of the things that you'll see is a very limited number of phenotypes or appearances. And so 2 examples of that are antibiotic-resistant bacteria and pest-resistant insects. So bacteria can be resistant to antibiotics, which is why it's so important that you finish taking antibiotics. So some bacteria have a gene that makes them resistant and they don't get killed by antibiotics. And some pests can have resistance to, or some insects can have resistance to pesticides. So that one single gene causes 2 different phenotypes. Either you're resistant to the antibiotic or the pesticide, or you're not resistant. Another example is lizard color. Here we've got a population with brown lizards and black lizards. So there's 2 different phenotypes. And so it's just controlled by one gene. We see within that one gene there was a mutation that caused a red phenotype, but that red phenotype was not very fit, and so it died out very early on. We can see this population is undergoing evolution because we start out with a lot of brown lizards, and over time the allele frequency changes, and so by the end here in generation 30 we've got much less brown lizards and we've got a lot more black lizards. So the allele frequency changed from our initial population to our population 30 generations later. But not everything is so simple in terms of phenotypes. Polygenic traits are traits that are controlled by more than one gene. And so kind of the hallmark of that, or how you can really tell is something controlled by more than one gene, is does it have a wide range of phenotypes? If it has a wide range of phenotypes, then it's usually controlled by many genes. So because it's controlled by many genes, each one of those genes has its own alleles, and so there's lots of alleles that contribute to the particular phenotype. And so it's the phenotype that natural selection acts on, not necessarily the genes. And so because individuals have a wide range of phenotypes, they also have a wide range of fitnesses. So for example, actually we're going to get to examples in just a second, but there's 3 different types of selection that can occur on polygenic traits. The first example is directional selection. Directional selection means that if we graft all of the phenotypes on a bar or on a graph, it sort of forms a bell curve. And so here we're looking at beak size. And there are a few birds that have very small beaks, a few birds that have very large beaks, but most beaks are kind of somewhere in the middle. And directional selection means that there's a shift in that average phenotype. So the dotted line was what we started out with. We started out with beaks that were a little bit smaller. And over time, the beaks got larger on average. So the smallest beaks became a little bit larger, and the larger beaks became a little bit larger as well. And so if the types of seeds that these birds were going after became larger, because there was an abundance of rain or new nutrients that were added to the soil, it becomes an advantage in terms of fitness to have a large beak. And so over time, the birds that have the largest beaks, they're able to get enough food to survive and reproduce and pass those genes on. So this is an example of directional selection. Stabilizing selection means that it's the average phenotype that provides a greater fitness than either extremes. So an example of stabilizing selection is the birth weight of babies. So it's a disadvantage to have a very low birth weight because that generally means that the baby was premature and it's not fully developed, and so it has a lot of health problems early on. It's also a disadvantage to be a very large baby. Large babies just can't make it out of their mothers, and so they're not born. They might die in the womb. So it's an advantage to be kind of average. So we start out with a wider range of phenotypes, and stabilizing selection causes the bell curve to get a little bit more narrow. And so we have more individuals that are average and less individuals that are on either of the two extremes. Disruptive selection means that the extremes, both extremes, large and small, are both advantages. So two distinct phenotypes are produced. So we'll look at a population of birds again and look at their beak size. But in this case, it's the extreme phenotypes, the smallest beaks and the largest beaks, that both provide equal and higher fitness than having the average beak size. So an example where this might happen is, let's say there's two different types of seeds on an island. There's very small seeds and there's very large seeds. Well, with a smaller beak, the birds were able to eat those smaller seeds, and larger beaks allow them to open up the larger seeds. If you're average, you're not really good at eating the small seeds or eating the large seeds, and so it's a disadvantage. And there's three other sources of evolutionary change besides just natural selection. The first one is genetic drift. So genetic drift is just a random change in allele frequency. It doesn't have anything to do with how fit an organism is. Just at random, the allele frequency can change. And so if you let's think about a really dramatic example. So imagine that you start off with a population of 2 individuals, and both of those individuals are heterozygous for a particular trait that's controlled by just one gene. So let's go back to our kind of classic example of flower color in peas. And let's say we've got 2 heterozygous peas that are both purple for their flower color. And those 2 purple flowers mate, and so if you work out a Punnett square, there's a 25% chance of those flowers being homozygous dominant, 25% chance of those flowers being homozygous recessive, and 50% chance of those flowers being heterozygous. Well, in the event that that 25% chance where you would have a homozygous dominant allele, or homozygous dominant individual, well, now we've just lost that recessive allele. That recessive allele is going to die with the 2 parents. So once those 2 pea plant parents die that had the white allele, if their offspring just happens to not have any, well, now we've changed the allele frequency in our population so that it no longer includes that white allele. So you're going to see genetic drift happen more dramatically and more often in small populations. One type of genetic drift, which is the second source of evolutionary change, is called the bottleneck effect. And the bottleneck effect is exactly like this picture. So we start out with a parent population, and this represents our gene pool. And for some reason, there's a drastic reduction in the population. So a very common way that this happens is through natural disasters. And there's no particular phenotype or genotype that is gaining an advantage here. It's just at random which individuals survive versus which don't. And so that's why we're showing this bottle just randomly pouring out a few of the alleles from our original gene pool. So these surviving individuals then are able to reproduce. And as they reproduce and form the next generation, you'll notice that this bottle has a lot more blue alleles in it than our original gene pool. So we see evolution here because we've changed the allele frequency from our parent population to the second generation because of a dramatic reduction in the population size. So natural disasters are common ways for this to happen, and also human causes cause the bottleneck effect, such as when we overhunt. And the last source of evolutionary change is called the founder's effect. The founder's effect happens when a new population colonizes, or a population colonizes a new habitat. And so if you imagine here, this is our original population that lives on the mainland. And so we've got black bugs, yellow bugs, and red bugs. And a small group of them gets blown over to an island. And so here's our group that gets blown over to an island. And just by chance, it happens to be black bugs and yellow bugs. And so none of the red alleles made it over to this island. And so because of that, you don't have any red alleles in this new population. So if we compare our old population with our new population, we can see a difference in allele frequency. This population had red alleles, and this population had no red alleles. And so this is just showing two examples of the same thing happening. One small population gets blown over to one island, and a second small population gets blown over to another island. And so you can see these two populations are very different after they've colonized this new habitat.